Hello, everyone, to another episode of the Omics cast. And today I'm going to talk about missing heritability, which is something that a concept that's come around the world of genomics for quite a while. There's been a couple of recent news on missing heritability. There's been a paper by Peter Visscher who with his collaborators, they found a way of extracting more heritability from whole genome sequencing analysis. We discussed this in podcast that we do called Cambridge Genomics, and you can search for it on my Twitter account. The last slide will give you the details. And then there's been a blog post by Alexander Young commenting on what Peter Visscher had found out. Peter Visscher basically said that if we, instead of genotyping with genotyping arrays that contain, let's say, 1 million SNP positions, if instead of genotyping individuals with genotyping arrays, we perform whole genome sequencing and we find out all the SNP and indels in each of the samples, that then we can find a lot more rare variation, and that these rare variants that are only present in a few individuals in the population, to the point where they can be even private, meaning only one individual in the sequence whole population of the world contains that SNP, that variant. These rare variants, especially the ones that are in low LD, in low linkage disequilibrium, are the ones that contribute the most to this missing heritability, and that when we move from genotyping to whole genome sequencing, we can find out a lot more heritability that up until that now it was missing. So this is what the paper details, and then Alexander Young made a blog post about it, revisiting this concept. So Alexander Young starts saying that heritability is a fraction of trait variation in a population due to genetic inheritance, and that this is something that started with Charles Darwin and Francis Galton, and that most of what happened there was in the last few decades was studying twins because if they're homozygotic twins, monozygotic twins contain the same genetic material, but they may be slightly different in their phenotype as they've grown up and evolved. And so we can find out what makes two individuals that have the same genetic content different, and find out what part of it is heritable. And a few decades ago, genotyping technologies, mainly the ones from Agilent, Illumina, etc., allowed the scientific field to measure hundreds of thousands of genome-wide genetic variants for thousands of individuals. I would say the mark now is probably at around 1 million, 1.5 million SNP positions, and the cost is, it can go in between $100 or even below $100 or below, maybe $50, depending on the size of the study, per individual. And so the question is, what happens with these variants? How do they associate to phenotypic effects? So this is the dawn of the genome-wide association study, GWAS, and a lot of software and tools have been developed to find out more about this. And so in this paper from Manolio and collaborators in 2009, they hypothesized where would these variants be and what would be their effect size. And in this plot, it shows this diagonal here of rare alleles causing Mendelian diseases. This is what the rare diseases field is all about, and common variants implicated in common diseases by genome-wide association. And then in the middle, low frequency variants with intermediate effects. But then there are two corners to this plot. The rare variants with small effect, very hard to identify by genetic means, meaning genotyping. And for example, high effect common variants influencing common disease. And we have some examples of this, right? So this is a plot basically summarizing the variants in allele frequency, the rare ones, very rare low frequency, common ones, and what their effect size could be. And this is a paper of 2009. And so Alexander Young in this blog post says complex traits are highly polygenic and affected by many rare variants. And if you do genotyping, you may miss quite a lot of these variants. And so in the Peter Visscher paper and recent papers, they said that they estimated that the genetic variation captured on the array, on a given array for a number of individuals would explain around 45% of the variability in height in the population. So that if you want to explain why you're a certain height and you compare it to the height of your parents and your grandparents, your great-grandparents, some of it is going to be nurturing what you ate in the first 5 years of your life, etc. But some of it is going to be simply what is the genetic makeup of your genome. And so you're going to correlate your height with the height of your parents. And if your parents and grandparents were tall, 45% of your height is going to be influenced by your parents and predecessors also being tall. There was in 2015 an improvement into the techniques used to do genome-wide association study, which was called imputation. And the imputation is basically saying if one of these spots in the genotyping array is not present, or if we know there are common SNPs in between two spots, we can impute what that missing SNP is by knowing what the state of the two spots left and right to it is by the genotyping, right? And then this increased the estimated heritability for height from 45% to 55%. So we can basically say we can extract more juice from these genotyping experiments, and so know more about the genome of the individuals that are genotyped, and then we can explain another 10% of the heritability for height. And then this is where we discuss the preprint from the Visscher group, and Alexander Young says that the paper claims a recovery of trait heritability for whole genome sequence data based on an impressive data set of about 20,000 individuals of European ancestry, unrelated individuals. They're not connected by a close family link. And they did whole genome sequencing data, and they covered 47.1 million genetic variants, which is a lot. So if you consider that each of us on average has about 2.5, 3 million variants between SNPs and indels, then doing 20,000 individuals and capturing 47.1 million genetic variants is a lot of data and a very good starting point. And then highlighted here says the analysis presents evidence that rare variants that are not well imputed, typically below the 0.1% minor allele frequency, contribute significantly to the heritable variation for height and BMI. BMI is the second trait, body mass index, the second trait studied in this paper with this cohort of individuals. So 0.1% minor allele frequency, it means only 1 in 1,000 individuals have that allele, have that genetic variant, and then the other 999 individuals don't have that one, right? So it's a very low frequency. Paper also presents evidence from an outsized contribution from low frequency protein altering variants, especially those in low correlation with other variants in the genome, low linkage disequilibrium, which means in your genome, as you go along downstream of from a chromosome, you can find a variant there that is not linked to the previous variant before upstream and not linked to the next variant downstream. It's basically a variant that popped out of nothing there. And for those that are low frequency protein altering variants, basically meaning they change the makeup of one of the proteins that is encoded in the gene where that variant falls, those low frequency variants, protein altering variants that are in low LD contribute a lot to explain the rest of the heritability that wasn't found beforehand. And then it suggests that the paper suggests that the full heritability estimated by twin studies is waiting to be unlocked by whole genome sequencing data. And so when we have many individual sequenced by whole genome sequencing data, it could be with technologies from Illumina or from BGI or from PacBio or from Nanopore. When we've done lots of these whole genome, produced lots of these whole genome data sets and study them collectively, we can find out more of this missing heritability. And then Alexander Young makes a series of points to punctuate or to qualify what this preprint from Peter Visscher is concluding. The first one is that these estimates of heritability have a large confidence interval. So a 40% with standard error of 9% implies that with 95% confidence, the heritability is between 22% and 57%. So it means we're 95% confident that we have a minimum of 22% of heritability explained, and it could go up to 57%. So it's a wide range for these studies. Another point made in this blog post: if a rare variant happens by chance to be carried by a few individuals who are outliers for height or BMI, very tall or very short, or very fat, very thin, then this could lead to overestimation of the effect of that variant. And that's basically saying it's a low sample size issue, not collectively for the study, but for those very rare variants that are carried by extreme individuals, that they are outliers for the trait that you're studying. And third, another point that he makes: the distribution of effect sizes among rare variants are therefore likely to deviate strongly from the modeling assumptions of the method employed. Meaning, the point here made is that the technology and the statistical analysis that you use matters, and that these effect sizes can deviate strongly from the assumptions in your model. For example, if you study different European populations, different humans from different European populations, in a mixed sample of Dutch people from the Netherlands and Italians, any genetic variant that is at a higher frequency in the Dutch than the Italians would be correlated with height, simply because Dutch people are taller than Italians on average. So what this fourth concern, population stratification, is about is that no matter how well you sample a population, there's always going to be differences from different ethnicities being stratified for whatever, in this case for height. And Dutch people are the tallest in Europe, whereas Italians are shorter on average. So those alleles that are different in frequency in the Dutch, they will look simply linked, even if they're not, they'll look simply linked to height because of the difference in frequency. So all that this means is that you have to compensate, you have to correct for population stratification, and there's different methods to do that, and the argument is that none of them is perfect, but some are better than others. So Alexander Young says the missing heritability problem has not been solved by this preprint from Peter Visscher, although it does represent an important advance in the detail of quantitative genetic analysis in what we understand from how variants in our genome affect traits, affect things that we see on our phenotype, our height, our BMI, the color of our eyes, the curliness of our hair, etc., etc., and also some that are clinically relevant or even more clinically relevant than BMI, body mass index. And so that's the conclusion from this blog post, which you can find in the first slide, the link of the first slide here. That's the blog post, and that's the paper from Peter Visscher. And this set of slides you can get a copy if you want by contacting me either via Twitter or LinkedIn. Here are the links for both. I hope you enjoyed this video cast. This is a video cast of a series where I talk about omics, genomic technologies, omics in general, and I try to explain some of these findings that are being published in social media, especially Twitter. Twitter is probably, in my mind, the best way, the best place to see what's going on in the field currently. That you can find out, and you can follow people that you're interested in, and then find out more about the results of their science. I hope you enjoyed it, and I hope you subscribe to the podcast, which is available at bit.ly/the Omics cast, all one word. Thank you very much, and I'll see you in the next episode.